All right, welcome to TIL Talks with Chris White, where we talk TILs, tumor-infiltrating lymphocytes, and all advanced cell and gene therapies, both with patients and caregivers, along with industry leaders. Let's get going. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. Hello, what's up? What's up? Welcome back to another episode of TIL Talks with Chris White. I am super excited for today's conversation. I have one of my repeating guests, Dr. John Powderly from Carolina BioOncology Institute and BioCyTIcs with me again today. And we're going to just dig in and pick up where we left off from our last conversation. John, give us a little 101
Thanks,
on where we're
Chris.
going today.
I like we're getting in the cadence about every 3 or 4 weeks. And so what I'm doing for your audience, I'm pulling out some old slides over the past couple of years and putting them in some teachable order so they make sense. And so we have a good 45 minutes we can go through and ask good questions. And I think that a lot of these slides will resonate with your cancer audience. The first thing is I will share my screen here. First, let me get it up here. So I've got good slides up. So at the last talk we did, yeah, you guys can see that now. So we talked about
Yeah.
what are the modalities? Modality is like a term for the treatments, right? So what are the different treatments for cancer in the broadest categories we know? And this is an old slide from 2005. And up there in the middle, you can see, okay, it's like a glass human body, and around it has these different types of treatment options. Up in the upper left is things that are in the red. I call them old school. These are old school things that really have been around 30, 40 years. And the upper left is plain old chemotherapy, right? And most people may not know, but chemotherapy is mostly comes from two sources. One is heavy metals. So platinum, people will hear the term carboplatinum, cisplatinum, oxaliplatinum. That is, it's heavy metal. It's platinum that is in the toxin. So that it can be given into the human body, and heavy metals can damage DNA, and it works. They work for aggressive cancers, but you do have hair loss, a lot of vomiting, and patients will say they can even taste the chemotherapy, the metal in their mouth. So you'll hear that a lot. Chris, I don't remember. Did you get a platinum? I don't remember.
Yes, I actually had a regimen of what they call CVD, which is three separate chemotherapies. It's cisplatin, visplatin. I'd have to go look it up, but
Doxorubicin.
yeah, but I did after I progressed off of PD-1 and CTLA of 2 cycles. My progression was pretty much out of control, and we went to the CVD chemotherapy to try to bring down my tumor burden. And they also requested to do a monotherapy of immunotherapy during the same time. But during this time frame, that wasn't actually FDA approved, so I was only able to get one dose. But I did go through 3 cycles of that, which was 3 days a week and that was those 3 days a week were 1 cycle. And so times that by 3. And then later on towards the end of my treatment before I did TIL, I ended up doing a different chemotherapy. But
That CVD, the cisplatin, vincristine, and
yeah, it's like
rubicin. That's a doozy. That's the big
it's a
gun.
triple banger right there.
They must have really had to get control. It's a sledgehammer, so I'm sure you lost your hair, you had the neuropathy,
I
nausea,
Yeah, it
mouth ulcers,
it
diarrhea.
hit it
That's a
it
tough.
it was a storm.
So the chemotherapy is a good sledgehammer when you need to get control. And we say, and every time you get a dose of chemo, the tumor has a log kill. A log kill, meaning that if you have, for example, a billion cancer cells, let's say that might be a golf ball to tennis ball size, billion cancer cells, every time you get a dose of chemo, in that 10 billion might go down for teaching points, 10 billion to 100 million. And then one more dose, you go down to 10 million. Another dose, down to a million. So it really has a good kinetic kill in general, the first few cycles. After the first few cycles, the kill is not as much, but it does get patients out of a pain crisis, all right, which is how we use it in the metastatic setting or when it's uncontrolled growth, like in your situation. And usually chemo these days are giving in doublets or triplets. So you got a good triplet of
Yeah.
different classes of chemotherapy. So the heavy metals are one class. The other vincristine or vinca alkaloid, that is another toxin comes from, I want to say that's a plant-derived one. And then the D, the doxorubicin, is from some bacteria or fungus. I forget which one. I think it might be a fungus. So the old-fashioned drugs are generally discovered from years of experiments of what in the animal kingdom or the plant kingdom or the bacteria or fungal kingdom has these toxins that have some cidal or killing effect. And so they were very, very effective in the 80s and 90s, and we still use them today to get logarithmic bulk kill when tumors are bulky. The bad news is generally, most oncologists will agree, they work great for 6 months or so, but then the tumors are smart. They know how to pump them out or become resistant to those drugs. So chemotherapies are still around. They're still kind of considered a lot of the front line or in even adjuvant setting for many cancers, and immunotherapy is being used with that. But yeah, the cisplatin you got, it's so toxic they have to give it over 3 days so it doesn't burn the kidneys. We don't even use that much for melanoma anymore. So it's amazing they got you got that, and then you went on to get TIL.
Yeah. I mean, at the time, it wasn't FDA approved to do it in conjunction with immunotherapy, but I believe it as of today, it is.
So, the other up on the upper left hand, these are again the kind of the older historical standards as a modality. Radiation, which has been around a good 40, 50 years, surgery, old fashioned surgery. Now, a lot of modern surgeries are robotic. Radiation is becoming more sophisticated with more and more narrow beams. Now we can aim a beam so narrow and tight, they're called stereotactic radiation or so called gamma knife. And then you'll see down there, endocrine therapy is used for hormonally sensitive cancers, breast and prostate. And all the other ones in green, I would say that those are the modern generation modalities, different types of modern treatment options, which are much more targeted. And we like to tell patients, we didn't discover those other ones; those are engineered. The chemo we discovered, then we engineered it a little bit. The toxins from the animal kingdom, those were discovered, and we kind of engineer them, synthetically made. Everything in green really are engineered to hit targets. And so going from the top right, the biologics were the first big class. So those were biologics mean they're made through a living organism, living system. They used to be made in bacteria, engineered in bacteria to secrete them, like the original genetically engineered bacteria where to secrete insulin was the first biologic. Then
Wow.
we got more classes of biologics. And those three there, the cytokines, cyto means cell, kine means to stimulate or to communicate with, both to communicate while stimulating. Cytokine is like a cellular communication. So in immunology, we might say an immune cytokine or immunokine. So cytokines in general, they communicate cell to cell, and a little bit they might spread through the body some, and give their signals. So the most famous cytokine in immunotherapy world that were first developed was the interleukins and interferon. So, which are the original gas pedals that we discovered of cytokines in immunology. Dr. Rosenberg discovered the interferon and discovered the interleukin. Interferon was a few years before that. It was discovered through viral infections and how the immune system might fight viruses. And I think you might have heard me say the front page of Time magazine was interferons going to cure cancer. And then
I was gonna mention that.
yeah, then the front page of Time magazine a few years later was interleukins going to cure cancer. And Dr. Rosenberg, my mentor, wrote a great book about the discovery of interleukin, which was how white cells, leukin means white cells, interleukin, how white cells communicate with each other through these cytokines that they secrete. Interleukin 2 was the one that helped grow lymphocytes. And he wrote a book on that called The Transformed Cell. It was one of the top selling bestsellers. It's a good history read. Probably came out in right around 90s, I think, when I was in med school, early med school. So the other class is growth factors. Those are used really to stimulate the bone marrow. And so after you got your CBD chemotherapy, you probably got some growth factors to stimulate the bone marrow. And growth factors would be one class is called GCSF. You hear that, which is granulocyte colony growth factor. There's other growth factors that stimulate the white cells, which is GCSF. There's growth factors to stimulate the red blood cells in the bone marrow so you can recover your bone marrow after chemotherapy. And there's even growth factors to stimulate the platelets as well. So the growth factors are commonly used around chemotherapy to boost the bone marrow back up.
Can I ask, do they provide those in a little device that they give you and they attach to you, and 24 hours after your actual infusion, it actually goes off and then, you'll hear a little click and then that, means you
Yeah, we're getting
received it, and
pretty
then
modern.
afterwards you take it off and throw it away?
Yeah, the first few growth factors that came out in a couple companies claim to fame, they first developed growth factors and they're now some of the biggest biopharma companies. But they first came out as single shots, like daily, you'd give them for 4, 5, 6, 7 days. So for the bone marrow recovery, then they learned how to give them as a depot shot. Depot meaning it would
Okay.
deposit in like an oily substance, so it would leach out. And now they're making it so when you get your chemo, they can put on a sticker, right, like a pad with a small needle that 24 hours later the shot will go in as a depot under the skin.
Yeah.
Yeah, so like a delayed trigger release that's has a timer on it.
That's what I remember from those chemo infusions was
So
that
that's a
every
big
time I would do that,
convenience.
they would give me one or put it on me, and then 24 hours later, I'd be at work doing whatever, and click, I'd be like, "Oh,
And it prevents your bone marrows from bottoming out. It'll make it so they
okay.
can give you the full dose CVD, right? Through 3
Yep,
cycles. If you
yep,
get a nice boost of the growth factors after each dose, it keeps your bone marrows adequate. And then
okay,
so yeah,
makes
so those
sense."
are very good support drugs, growth factors for heavy duty chemotherapy. Whereas the cytokines are more immunotherapy based. The chemokines means that the intention there is how do cells attract one another, right? It's signaling, but how they track and how they will climb through basement membranes or how they will signal to each other to change into something different, like change their appearance or their function. So it's really more about signaling kinds. Peptides are kinds. Peptides signaling that make them have chemotaxis, which means I'm going to follow that trail of where the signal is the weakest, and I'm going to follow it to where the trail is the strongest. So that would be an example of a chemokine. So chemokines are valuable. There are a couple chemokines I think in research immunotherapy. Sometimes you block a chemokine. You can block that pathway if you don't want cells to migrate in certain directions. Like there are some chemokines now that are blockers that are used as immune suppressants, right? So immune cells don't track to the inflammation. That would be good for autoimmune disease. But if you wanted to develop a chemokine to help cancer immunotherapy work better, you would try to create a drug that would secrete chemokines to attract white cells to follow the sniffing, the trail up to the higher gradient of where the strongest scent, if you will, of the chemokine came from. So you would put a injection of chemokines in a tumor so immune cells could track there.
Okay,
So
that makes
just
sense.
as a class, so people can hear these words. Antibodies, you hear the term monoclonal antibodies. Antibodies are natural immune proteins in our own body that we make in our own immune cells, which we have in our body. We call them immune globulins. People have heard, what's your titer? What's your immune globulin? Those are made up of antibodies, and every antibody is part of your immune system that is naturally trained to fight off viruses or bacteria or infections. So what scientists did about also in the 90s is begin to learn how to engineer or manufacture an antibody against very specific targets. So that's where at the tip of that monoclonal antibody, you can see like the yellow. That means that yellow is how the peptides have been sequenced exactly right to recognize something like the top of a tumor or to recognize oftentimes the top of a tumor. So it'll target the tumor. So monoclonal antibodies and some examples of monoclonal antibodies. The first one that came out was the famous drug rituximab. I'm saying the scientific name against the
A lot of blood
lymphocyte, the B
cancers use
lymphocyte,
that, right?
lymphoid B cell lymphoma. The next big famous antibody that came out right around that time was for women with breast cancer, Herceptin. Well, I can't say Herceptin; it'd be hertuzumab, right? That's would be the scientific name, not the brand name. But the other monoclonal antibodies that are claimed to fame are drugs called bevacizumab, which are used to block the growth factors that stimulate blood vessels to grow around a tumor. So bevacizumab is one of those drugs that came out also in the mid to late 90s. Big impact on cancer treatments to cut off the blood supply around the tumor with monoclonal antibodies. The way you can know if it's a monoclonal antibody is so if the drug name has MAB in it, MAB, MAB, you know it's a monoclonal antibody. The
Okay,
other trick for the audience to know is if a drug ends in iximab, like rituximab, the first one, the Ix means it came from a mouse. It's a mouse antibody. So the mouse is the system on how the antibody was first made and learned to fight that target. So the Ix means chimeric for mouse origin. A
so from,
rituximab,
like, early trial
the
stage
the
from
first
mouse
2
models,
drugs,
or
yeah, the first 2 drugs like rituximab was about 69% mouse. So when the FDA goes through the naming convention that's harmonized across the world, the X mab is monoclonal antibody. The X in there, the iximab, the X part of the X is a reminder, dear doctors, this antibody is part mouse, therefore they might have bad allergies. Be aware. So it's an extra precaution that the patients may need diphenhydramine or Tylenol or steroids before
Okay.
prevent allergic reactions. Another example of a famous antibody, speaking of the naming convention, is if a drug ends in izumab with a Z, like izumab, like bevacizumab, the Z means humanized because there's a Z in humanized.
Okay.
Drugs that end in Z, izumab, are 99% human and only 1% mouse. So we say they're humanized. So they're less allergy prone,
Okay. Okay. Okay.
generally less reactions. And then if a drug ends in umumab, in which the first umumab that came out was ipi and panitumumab, where I think they were kind of tied. Technically, it was they were very tied, like about a couple weeks apart. But yeah,
Close.
they end in umumab, which means they are fully human. When to make a human antibody that actually gave a human's immune system to a mouse. They engineered a mouse to have a human's immune system, at least the B cell compartment. And then they would inoculate the mouse with what they wanted to create an antibody against. And then they found that immune cell or the B cell that mounted that antibody, and then they cloned it up and figured out the gene. So it's very elegant how there's two companies in the very early days in the
It was
mid
like
early 90s.
2011 or
Yeah.
something when
That's
it
when it
finally got
became approved,
approved. Yeah.
but when it
Okay.
was being developed, it was like early to mid '90s, and two big East Coast, West Coast companies were the first to do fully human antibodies. They both got one on the market right around the same time for cancer, ipilimumab,
And that was early 90s?
and then panitumumab, which was for epidermal growth factor, like colon cancer for panitumumab. So that's a good background there. The radioimmunotherapy is kind of becoming more popular now because they're taking antibodies, monoclonal antibodies, and tagging it with a radioisotope or a hot metal, if you will, a radioisotope, radioactive substance, so that when it targets and the steering wheel through the antibody targeting will go to the surface of the tumor, bring in that radioisotope, a radioactive substance, a heavy metal. Yttrium would be an example of that. Some of them they're all over the map. Almost many heavy metals that you can think of have been turned into hot radioisotopes, and can be tagged on an antibody.
Gotcha.
What's not on here, which because they're kind of more recent, is a class of antibodies. Speaking of antibodies, a class called antibody-drug conjugates or ADC. And that is where we've got the immune cell. I'm sorry, you've got the antibody, the monoclonal antibody, as the steering wheel, and the tail of the antibody, which normally would recruit other immune cells to target a tumor. On the tail of the antibody, instead of just being a naked antibody to recruit more immune cells, they'll add in a toxin. And when you add a toxin, we call it an antibody drug conjugate instead of a radioimmunotherapy, not a radioisotope antibody, but an antibody drug conjugate or ADC. An ADC is a class of immunotherapy right now, which is very, very hot, every major drug company has ADCs in their pipeline. So there's probably been over 100, 200 in development, another 100 like in active phase 1, 2 studies, and maybe about a dozen or so that have been already FDA approved. In
Wow.
that class of an antibody, the nice thing is it has the nice properties of an antibody. It's targeted, it hits the surface of the tumor, it has some immune attack benefit, but that toxin is so potent as like a single molecule, like a single potent like chemotherapy molecule. Sometimes they'll use chemotherapy that was so potent they couldn't even develop the chemotherapy anymore. It was too toxic. So then they said, "Oh, let's go back. Let's take that off the shelf, get it out of mothballs, and then take that really toxic, super toxic molecule and tag it to the tail of an antibody. Therefore, it's not just widespread in the human going nowhere. It's now in the human going directly to the surface of the tumor with that incredibly highly toxic, potent molecule.
So
And they'll
it's
tag
more specifically
3 mo
targeted, right?
so it's very targeted. So antibody drug conjugates, the nice thing is they're targeted. They have the steering wheel of the antibody. They bring a little bit of an immune component because they still have a tail, right, to recruit immune cells. And then they also bring a toxin. So we tell patients, dear patient, this antibody is going to steer to your tumor surface, hopefully 99% of the tumor on the surface rather than the rest of the body. And then the toxin will cause an inflammatory death, right, in the tumor that recruits more immune cells, and then 3 weeks later, they get another dose of the ADC, antibody drug. So those are a good class, lots of trials right now. I think at our clinic, we've got at least 10 of those good trials open right now. So down at the bottom, now we're getting a little more targeted with antisense is a term that means gene silencing. So if there's a gene in your body that's driving your cancer, like let's say people might hear, the early days we had a gene we were targeting, the first antisense that made it into cancer was a drug targeting BCL2. BCL2 is a apoptotic gene, and if you can block it or silence it, it makes tumors more sensitive to die. So that was used actually in melanoma. Chris was an early trial for melanoma to create an antisense, which anti means it was the opposite strand, the opposite sequence of the DNA or the RNA strand. So it would bind to the actual RNA strand and silence it, and therefore that target within the cell, now we're inside the cell, not on the surface with an antibody, but inside the cell, you could silence or turn off genes inside the cell through antisense. So those were first done in like that was probably late 90s, early thousands, where those studies started getting into humans. And now fast forward, we do have antisense drugs that are in other indications, not in cancer yet. I don't think any, but their antisense drugs are being used in other indications, not cancer.
So 20 plus years in development though, right?
And yeah, the
Yeah.
tricky part with antisense is antibodies are kinda they're large molecules, and we put these into other classes. Large molecule means antibodies. You talk to scientists, what are you doing your R&D in, or what's your biotech company's angle? "Oh, we're large molecule." "You mean antibodies, large molecule." And then antisense is its own like camp of antisense or gene silencing, or mRNA might be in that camp. And then there's the small molecule camp down there on the right, which they're targeting, they're using small molecules, not chemotherapy, but trying to engineer a very foreign looking molecule that doesn't exist anywhere in nature. They'll engineer it so that it will target a very specific pathway inside the cell. So the modalities that work inside the cell are the antisense, because that gets into the genes inside the cell to silence a gene, or small molecules work inside the cell. And chemo works inside the cell because it knocks down your DNA. Endocrine therapy works inside the cell because hormones actually get inside the cell to signal them. But those other treatments there, the cytokines, the chemokines, the antibodies, the cellular, those all work on the sense they work on the surface of the cell to control the cell from the top and hopefully destroy it, but they use the surface. So kind of different. Even the vaccine, you might give a vaccine, it stimulates the immune system, but where the immune system goes around and looks for things on the surface of the cell to attack.
Like the mRNA vaccine?
The mRNA vaccine, so technically the mRNA vaccine, this is exciting stuff. The mRNA itself, for it to manufacture the protein, which is the vaccine, the mRNA goes into the cell like the antisense, but instead of blocking a gene, the mRNA kind of creates a new transient transcription of almost a gene, not in the genome, but near the genome, to create a new protein. And the reason for the vaccine is to create a protein that might be recognized as foreign, that the cancer might also have, so that your immune system will start to mount a more vigorous reaction against the vaccine, and then as a bystander also attack the tumor. So vaccines were kind of really popular in the early 90s, late 80s. I think it's kind of jokingly that the largest NCI budget ever spent for one single research trial was a West Coast, the John Wayne cancer vaccine trial, using some whole cell tumor vaccine. They would grind up tumors, it'd be a whole cell vaccine. And because that's all they had back then, grind up some tumors, make a whole cell melanoma vaccine. It was for melanoma. So if you can stimulate the immune system through brute force, grinding up a bunch of,
Yeah, use
and
what you got. That's all
that
they
was
had at the time.
big question, a big trial, a big thing, and, it turned out it didn't really work. I think in retrospect, when we look back and we thought the reason it didn't work is back then we didn't know the breaks of the immune system. We didn't know the CTLA-4, we didn't know the PD-1. So now that we've learned that, vaccines are starting to come back again, not as a wholesale ground up, cancer vaccines from grounding up other people's tumors and giving as a vaccine. Now cancer vaccines are coming back more of a purist approach. Purist meaning just giving the peptides that are the most antigenic or most foreign, the peptides themselves, or giving an mRNA into your body as a vaccine. So then the mRNA can be turned into the peptide in your body and to cause the vaccine that way. So
Okay.
those are they're making a comeback, because now if they're given in conjunction with checkpoint blockade, the immune system is likely to have a bigger response than we thought in the 1980s. So those are just making a comeback.
Gotcha.
And then we talked about angiogenesis. That's a modality of cutting off the blood supply around a tumor. And some of the antibodies can do that. There are some small molecules that'll focus on cutting off the blood side, but that's an important modality. Turns out that tumors like to grow, and tumors also know how to steal the blood, just like they know how to steal nutrition, right, from patients. They know how to steal nutrition,
That they
they know how
do.
to steal a blood supply. And if you can cut off their blood supply, you can really make a dent in tumors' growth. And there's this big class of drugs targeting anti-angiogenesis or blocking the blood supply. So angio means blood vessels. Genesis, like the growth of blood vessels. So anti-angiogenesis is you're blocking the growth of new blood vessels, right? And specifically around cancer.
Essentially, like stopping like the food supply
Yeah, yeah, there you go.
for it to grow on.
Most of those classes of drugs, this is from 20 years ago, they still are not the chemo. Nobody bothers making chemo anymore. But for the most part, the biologics, the small molecules, the antisense, the gene therapy, the cyto means cell, cytotherapy, the cell therapy, the vaccines, these things are still the very much part of the modern pipeline. Just everybody has their own version of what they're developing, but this is where we can say today there's like 9,000 new drugs in development. The pipeline's hot. They're all going to fit into one of these biological classes, if you will.
Got
So, all right, good. So let's see. So we'll go here. Next slide. So now we're gonna kind of switch gears and talk about. So that last slide was kind of the final slide of the last talk. Now we'll talk about this a little bit more closer to home. This is a talk I gave about a year ago down at a North Carolina, South Carolina, and Association of Community Cancer Centers um, symposium for community oncologists to get their continuing medical education and have conferences. And they asked me to give a talk about how to build a phase 1 program. And you've heard me talk about this. It's a little bit more like for the docs. So what would I be telling doctors, oncologists? Hey, if you were going to try to build a phase 1 program, what matters? This last talk, we looked at it from the patient standpoint. Dear patient, why would you want to be experimented upon? Dear patient, why would you go seeking a trial? What matters to you? So now I'm switching gears and saying, okay, dear doctor, hey, why would you want to start a phase 1 trial? Like,
exactly.
oh, man,
Yeah.
I got enough on my plate. Or dear hospital system, why would you want to start a research program? What's in it for me? What can I do? So this was to kind of get them interested and involved in maybe considering the risk benefit reward or the value proposition, right?
Right.
So it's that's where the angle's coming from. So your audience will find this kind of interesting. So first is the general outline, and hopefully we might get through half of this in the next 30 minutes, and then but the outline is what is the cancer research pipeline? Okay, what's the pipeline? How much does it cost, right, to develop drugs? This is just teaching doctors. They didn't learn this stuff in med school. A lot of the oncologists learn this through osmosis because
Yeah.
have to know about it. They're engaged in phase 2 and 3. Their hospital system has a program. A little bit about how broken is the clinical research system. Why build a phase 1 program? Like, who cares? Like, does it matter to your community? I don't know. I mean, does it matter? You tell me. Would your community want this? Okay, what is it? What is the phase 1 program? And then you think in the business mind, what are the deliverables? What do I need to do? What is it? Is it a widget? What am I delivering? Is it a service? Is it a product? What's the deliverable? And then do patients benefit? Why bother? Is it worth it? We're going to get deep into these. I got like 10 slides about each of these topics. How does it operate? How do you turn it on? What type of people do I need to hire?
Yeah.
Like, I'm leaving this.
Infrastructure.
This is too much. I need a drink. They're like, get me out of the room now. They put me on at the last day of the conference, by the way.
Wow.
No slight to me.
They're like, here, John, close it out.
Hey, I'm lucky I got invited. I'm lucky I got invited.
But this is all important stuff,
This is important
that
stuff.
upcoming
But literally,
oncologists
that's
need
what
to
happens.
know.
Research trials are often the last thing doctors think about and the last thing patients think about.
Which
And
is so opposite.
last program at a 2-day conference.
It's so opposite because literally, say they start the conference or symposium, whether it's a multi-day or 1 day, with that starts off with the why, the purpose behind it all,
Bingo.
and it
Yeah,
gets
yeah.
everybody fired up. And
Yeah.
we
So we
need that.
kind of know that because we're a little bit business minded. doctors don't really think about the why. They're like, the why is I'm here to help people. I'm here to follow cookbook medicine. I want to do a good job. I have to, I want to cater. But when you take a couple of steps back and say, why change to do something different? Look at the marketplace, the industrial complex. How can we fix it? How can we create maybe a different revenue stream of different quality of life for me as a doc if I want to do more translational research?
A
So yeah,
lot of
so
logistics, I think, that
lot of logistics, a lot
go
of
into
red
that.
tape,
Yeah.
regulatory. And then down at the bottom, speaking of that, like looking at the regulatory compliance challenges, the FDA inspection risks, where we have to be audit ready. What are the shifting markets? What's going out there in the markets? And then the last few slides, is it feasible? Can you sustain a phase one program if you do it right? We kind of joke, you can't do it as a hobby. It ain't gonna work. It's not
Yeah. You're gonna figure out the hard way
yeah,
on
you're
how
gonna
it really
learn, yeah.
works.
This is like all in. You gotta have a dedicated person. If you got like a 6 doctor practice, one person, this has gotta be their full-time job.
Oh, it's gonna
All
hurt.
right, we'll get there. So I forgot if I showed this slide to you before.
No, no, I would remember that, like, you
So
have not.
this is basically the math. Like I'm business minded. I'm like, okay, how much money do we spend of the US healthcare dollar on research for cancer? I'm breaking it down so I can make some assumptions here. So every single one of these has the link at the bottom as to the source. So this is not my opinion, which I love to talk my Irish opinion,
Yeah,
but this
right.
is looking at it, you step back. And this is 2023, like time period. Everything now just up at 15, 20%, right? So look at the healthcare dollar, upper left. The healthcare dollar back in 2023 was $4.2 trillion, which is it's usually about 20% of the GDP. So you take what is the GDP? 20% is healthcare dollars. Like that's like how much we spend of our GDP is on healthcare. I always thought a lot more will be spent on other things, but this, as a side note, most of what we spend our GDP on is consumerism,
Yeah, I
products.
agree. I agree.
Like so 20% is healthcare. That is a lot. But when you look at how much we spend on like buying stuff as like retail and consumerism, that's like I think 60%. It's over 50.
Yeah, it's
Um,
ridiculous.
so next is then you look at over on the right, top right, how much do we spend on all industry R&D, energy, petroleum, aerospace, engineers. Estimate there's about $940 billion on all R&D. And if you narrow that down just to the biopharma industry, which is 6.3% of all industry, this is 2023. The quotes at that time, which was the R&D industry, the biopharma spends about $60 billion per year as their annual budget. 60 billion is spent on cancer R&D. And I think I showed you a slide. I got a slide where you think that's a lot. They spend more on
I
more
believe it.
marketing than the R&D. Okay, and that's well borne out in other literature.
And the crazy thing is this is just a 3-year-old slide. Like, if we go back 6 years, those numbers, I mean, oh my god,
Yeah, yeah. So
what a
so this
dramatic
has really
change.
gone up. I had this same slide from 2005 when I started the clinic, and this is like 3 times more expensive than we started the clinic. So let's keep looking at these right and left. So
Yeah,
then we look over on the left, how many new cancer patients are there in America per year? Like 1.9 million new cancer patients. Okay. And all patients. Now, and you look, okay, of all those new cancer patients, how many participate in a trial? It's really like 2% participate in a trial, but we'll just round it. Quotes are 2 to 3. Let's round up 3% of cancer patients per year. So that's 58,000 cancer patients participate in a trial among the 1.9 million. Okay. Then let's look over on the right now. How many medical oncologists are there in the United States? There's 15,000. Usually we just quote there's 600,000 doctors. Maybe it's like 700,000. But how many are actually oncology boarded? It's 15,000. So we're there. It's a solid. It's still not enough. Then of those 15,000, how many are an investigator, principal investigator on at least one cooperative group study, which is because that's the low hanging fruit. That just means like your hospital system has one one federally funded research trial, just one that's federally funded. And that is only 13%. Then when you say,
yeah.
okay, what
This is
about
just a
like
fraction.
bingo? You say, okay, what about the number of oncologists that participate in an industry trial? which pays better, but it's a lot more complex because there's more at stake and because the biopharma companies have a much more bigger expectation that you're going to be
Yep.
engaged and involved and you're going to accrue the slots that they work to open up at your site. So about 1,000. And then we look, okay, of all those principal investigators, approximately how many do like phase one, like first in human, kind of as a at least some phase one. I put about 100. It's probably a little bit less that do just exclusively phase one as like a career move. I would say at every major academic center, it's probably one or two docs that do phase one as their job, or large tertiary academic or med center, large tertiary private hospital med center will do phase one studies. So that still is really around 100 docs that are like me, who I can compare notes with. We kind of
And traditionally, like, our phase 1 trials, usually the accrual is, I would just say on average, like a tenth of a later phase trial
so good
for
question.
the
So
accrual for patients. Is that correct?
remember, all new drugs have to go through phase one. So phase one is kind of like a bottleneck in that sense. All studies have to start at phase one. But the difference is phase one, because you got to
Safety, efficacy.
figure out the right dose. Phase one studies, though, there's maybe only 30 to 90 slots because you have to slowly escalate the drug. Remember, we talked about that last time. You can only enroll 3 patients at a time during dose escalation. And then
Accrual can be over a longer period of
18
time.
to 24 months, yeah, and that's in one of my slides that we
Yeah.
but yeah, it's slower, so that is a bottleneck, so there's not enough phase 1 docs like me. There's not enough trial sites willing to do phase 1, which are heavier lift because it's more complex. When you look at 9,000 cancer drugs in the pipeline, there's just not enough oncologists, not enough, like, system support, everything from just billable, like, there's no Medicare code for clinical trials. You know,
Yeah.
insurance companies aren't going to go paying anything for the trial part. They're supposed to pay for the support of the CAT scan. So a lot of this falls on small, medium biotech. Okay. And
Yeah,
so
and
small, medium,
that adds up, and it's
that adds
hard.
up. A small biotech, hey, they got $10 million for one drug and they're a new co. That'll barely get you through a phase one, plus all your other things, and keep your doors open for the next 2 to 3 years. So you take so that means a new drug with a new company that's spinning out and trying to do a phase one. I mean, they really have to raise 50 million just so they can have the infrastructure to manufacture the drug at a manufacturing facility, the regulatory part, their leadership team, their scientists, at least 10 people in that, and then to sponsor a trial. So these things add up very quickly. So this is why there's a lot of drugs in the pipeline of pre-IND or early research trying to get into phase one. And then there's a bottleneck just of which drugs are chosen for phase one, what small biotech can raise money to do a phase one. And then the phase one docs have a lot of business. So that in and of itself is like a bottleneck. Because the system is kind of broken around that. And we'll get into more of the reasons why through this talk.
Absolutely.
So then when you go back to the left and you say, okay, we know that true, reference facts, 25% of cancer trials close early due to low accrual or idle trials. So when you look at those numbers I just talked to you, you do the math, right? Just some basic math, and you say, okay, 60 billion of biopharma R&D dollars, but only 1,000 principal investigators that do like all phases across the US. That means we are spending, as a United States of America only, we are spending $60 million per principal investigator. All right. So just for that doc could like take some time away from their busy day-to-day chemo and standard of care and commercial billable insurance reimbursed, like to
It's like their
focus.
side gig, essentially, is how you can
Yeah,
right?
that's a lot of that's like a startup biotech company every day, $60 million a day to try to get the pipeline on. So we're not the thing is we're spending all that money to try to get drugs to patients, but the system is not working. The traction is not working. Now let's break it down. On the patient side, if you look at how many patients participate in trials, not just phase one, just all patients, based on the 60 bill of the R&D dollar, that means for each patient that goes in a trial, we as America, mostly the biopharma industry, is
Yes.
spending $1 million per patient per year. So you look at Chris, you were on a trial. Yours is a lot more expensive because you had the highest complexity of a cell therapy. But anybody that goes on any trial based on these average numbers, that's $100, it's $1 million per that patient per year. So I said, patient, I'm spending this. America as a biopharma research industry is spending more on you than probably you are getting spent on with your chemotherapy and
It's
other drugs.
true.
A little bit more. So now let's break it down a little bit more. We say the industry, if we're only accruing a quarter of the patients, we are wasting 15 billion industry dollars per year due to low accrual, which is equivalent to $41 million wasted per day due to low accrual. Breaking it down, if you look at like the blockbuster drugs, putting this relative to a blockbuster drug, we are wasting on idle trials, not accruing in the broken system, 1 million industry dollars per day wasted due to low accrual for each blockbuster drug. Each blockbuster drug. So you can see the weight of the system to try to make sure we can get eligible patients in the door, the efficiencies. And here we are, you and I are talking about, oh, we're faxing records. We're trying to get records from 5 hospital systems, trying to get patients earlier who are strong enough and screening through all that. And so this is why drugs are so expensive. $1 million wasted lost per day per each blockbuster drug because of the broken system. And so this is exactly one of the major reasons drugs are so expensive.
If they want to retain their money back, it's like a bank. You got to get your RO.
Exactly. They have to make it back. I understand. They have to. That's everybody deserves to get a return on their investment. But if we make the system more efficient, then everything becomes more affordable, and then the drug everybody wins. So just put that into perspective as kind of like the prelude to telling docs. Okay, here. So next slide here. So the cost of drugs. So this is a publication from 2023. Good timing, same year, on what is the average or median cost of developing cancer drugs, divided into 2 categories. One category is those where we're not looking for a companion diagnostic, like to see if the drug hits target with an assay or a marker or a biomarker. The other where it's a targeted drug, but we're just giving it willy-nilly. We're confident it's going to hit, but there's no biomarker assay no companion diagnostic. I just put it into 2 categories. Pretty much they're almost the same. But just to show you, the median cost of drugs is $2.6 billion and $3.5 billion for the companion diagnostic or non-precision. For all those cancer drugs that made it into phase 3 trials that went on to the market later, in that period of time. Now the range, look down the bottom. Some of those drugs, and we did the phase 1s for some of those drugs, many of those drugs. Some of those drugs cost R&D expenditure, at the bottom, that's the x-axis, upwards of $13 billion per drug. And on the right lower, it's $15 billion for that one drug. So the very wide deviation in there, but this is just telling you the real numbers published in real Journal of Pharmacy Policy Practices, how much biopharma industry, and ultimately, we as stakeholders, the Americans pay for all this. And then it comes back, and that's why drugs are so expensive.
It adds up. It's intense. Oh my God.
So now let's look at the hope, look at the promise. 9,100 cancer drugs in development. And the blue versus orange here is just looking at from year 2023 to 2024. There's so much R&D dollars, so much investment dollars being pumped into the development of new cancer drugs. And that the cancer drugs are at the top. On the left is the cancer. All the other ones are like non-cancer, neurology, infection,
Autoimmune. So yeah.
So cancer drugs is still the most highest profile, highest dollar spent. And that's a pretty big pipeline.
Oncology, it looks like it's almost at least double what it is compared to any other
right. Yeah.
disease or
Totally double. You're exactly right. And now we look at, let's just narrow it down to cell and gene therapy, since this is TIL Talks. This is focused on the cells, right? When you look at all the cells and gene therapies that are in development, based on this reference here from last year, 2 years ago, 2024. On the right, 2024, 2,100 in development. And to get those into a trial through phase 1, the phase 1.
And that was 2024?
That was 2024. Yeah, 2024. And it broke them down into cells and genes. So really we could say if you add cell and gene together, that's 4,000 of cell and gene therapies combined. Massive pipeline, that's a lot of people's 401k money, the small biotech.
Yeah.
That's a lot of hope.
Dude.
a lot of money spent, but the cell and gene therapy, they're just more complex. They need higher manufacturing complexity, higher phase one site complexity in teams to manage the side effects and all that. So just this is just to point out that this is real. And I think we talked about this, how disintegrated the system was. I forgot if I showed this to docs. So this is what I call horizontal disintegration. So this is looking back in um, 2005. This slide is from 2005. And as we started the clinic, I said, okay, what's the problem I'm trying to solve? I'm starting a clinic. I'm going out on my own. I want to create a research mission for a research clinic. The problem is that the cancer research system is broken. And I tried to outline how broken it is based on these disintegrations. These would be called parallel trades, where they're trades that are broken apart. Another way I would explain this is in the late 90s and early 2000s, it was very vogue to outsource everything. It's like America, everybody, all the companies started outsourcing. So the drug companies outsourced the manufacturing of the drug. They outsourced the research trial coordination. There's a lot of outsourcing. So there's other layers in here. But this was when I started my research clinic 21 years ago. This is what happened to the tumor up at the top. The tumor, you see the picture, the surgeon would resect it. It would go in the bucket, which is, formaldehyde or paraffin wax, not like in an incubator to grow it. It would just go in the bucket so that the pathologist could put it in wax or formaldehyde um, to go on a slide, paraffin wax on a slide. So they could look under the microscope, make a diagnosis. And then those slides would have to be saved for like 7 years for medical legal reasons or diagnosis. Then ultimately, back then when I started the clinic, most of those paraffin slides ended up in the trash. And any leftover tumor ended up in the trash. And so
So
this was what
7
happened.
years? They would have to save it for 7 years?
Medical legally, most pathology groups, most hospitals pathology groups will save the slides for at least 7 years. Usually it's 10 years. But they don't have the room to save it for 20
I
or
was gonna
30 years.
ask. I was like, where do they have the space to store all of that?
But they certainly don't save the bucket of the trash.
Ah,
That goes to hazardous waste incineration.
okay.
But the slides, they keep the slide. They keep a little paraffin wax. We say it's an archived block. So when patients go on trials, you hear them say, "Oh, we want some of your archived block from 3 years ago when you were diagnosed." They want to look at that sample, but it's not a living sample. It's just frozen, not frozen. It's in paraffin wax, so it's stuck in time in paraffin wax. So you can look at genes, you can look at proteins in it, but nothing functional about what's actively growing or moving, or from an immune standpoint, not what's being targeted.
Essentially, like, frozen in time, you can look at the
Frozen in time,
history of it,
but
but
not liquid
nothing.
nitrogen frozen, just frozen in wax.
Okay,
It's
that makes
it's
sense.
embalmed. It's like formaldehyde. So it's still rich data, but not very functional if you're like trying to say, "Hey, can you mount an immune response against my tumor?" No, not
Not
compared.
from 7 years ago,
No,
I can't.
your immune cells are shot. You've had all your chemo. Your immune cells it's hard to do that. So
Yeah,
part of the problem we're
absolutely.
trying to fix is we need to grow the tumors at the front end in culture. We need to expand them, grow them, expand immune cells and the tumor, mount these battlefields like in a functional assays, battlefield killing assays,
Something that's modernistic.
more modern. Yeah. Or so we can grow TIL. I mean, 0.001% of all tumors are like being frozen in liquid nitrogen, so they're viable and can be thawed, or being put directly into culture. So that's a huge one of the
Absolutely.
problems we're trying to address.
Absolutely.
So the clinic, it's got a nice door. I like to refer to the clinical door. The investigator, he's a doc. He's also called an investigator because he's doing trials. He might dictate what's happening. It gets typed into paper documents, and they treat based on what's called compendia, which would be like, what will the insurance companies pay for? Now we use the NCCN guidelines as our referee. Back then, insurance company would only pay for and what the pharmacy compendia would cover. Now the NCCN guidelines has taken that over. Patients would then sit in a chair, and then if you were lucky enough to get out of your paper-based records, you could actually use an electronic medical record and type in, which electronic medical is their glorified word
I
processors. They're
mean,
just archived.
electronic health records
It's
would
electronic
be
text
great.
documents. Yeah.
Open
So it's
air by
it's open. Yeah. So we're hoping to make it more structured. Like the other mission is like, hey, dear doctors, why you blah 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 blah? Let
why
me
are
teach
we
you
why
about
are we in the
what.
paper dimension when
Yeah,
we
why
could
not
be
bring this blah blah.
why could you be analog when you could be
Yeah,
digital?
why are you even you're using modern digital text-based, word processors, but let's figure out what are the data elements for a data model and pull out those data elements. So that means you have to say it. You have to say mild or moderate or start date or end date or attribution, quality, and all the metadata that goes with that. But that you can, so you can see where I get my mission is because these are the problems. So
I see it, and I just wanted to ask at some point, I want to be able to take this slide and let's make an animation out of each one of these line items from tumor,
yeah.
clinic, site management, and be able to.
So back then in 2005, when we started the clinic, trials were happening, but docs were too busy. And so there was two middle layers in there. One's called a site management organization or SMO, which means they're like, dear busy doctor, we know you don't have time for a trial, but we need you to be the investigator. But so we'll make it easy for you. We will do all the regulatory documents, the 3-ring binders, the drug accountability. We'll even help you get the special freezers with the temp tail and the monitoring and the case report forms when you enter the results of the data element of the side effects and all the trial data on paper. Or back then when that old computer looking, that
Sounds
electronic,
like a lot of extra work.
a lot of extra work. So then we upgraded from paper case report forms into electronic case report forms, which is another computer system. So you can see there are 2 computer systems: electronic medical records, electronic data capture. Totally separated, different data silos, different purposes, different profit drivers, different owners of that data. One patient, one doctor, different systems. It's still today, that's how different, disconnected that is still today.
Makes so much sense.
Then there's another layer called contract research organizations, which are great and they're important. There's always got to be good industry support to help develop drugs. A lot of these CROs, they do the heavy lifting, they do the work for the biopharma companies who are the sponsors, so that trials could be brought to research sites like ours. And these CROs, they're very, very very, very good at knowing how to open trials quickly, go to sites, get through contracts, budgets, and then show the monitor. They're bringing they always dress up nice and they bring their little
Right.
dolly because they're like the auditors. They're like the cross checkers. We call them clinical research associates or CRA, and there's certifications for them. And that's an expectation that the FDA has of a sponsor, a biopharma sponsor. Like on your trial, your biopharma sponsor had to do the trial according to the FDA requirements. I'm
Yep.
sure they hired a CRO. Everybody does. And that means a CRO person carried a little dolly and came to your hospital system and cross-checked and it had a lot of
She
queries.
was there when I signed my consent. She was there.
Well, that would have been the
She
coordinator. That
was
would have been the
local.
site. The
Yeah.
site would have been the site management. Now, the hospital system has their own, they are their site management. But out in the community setting, this would be another layer. But yeah, the site
Okay.
staff, the site team, the coordinator, they work for the site. So they work under the doctor that pays them. They're employed by the clinic or the hospital.
So,
They consented you.
like my CRO, she worked for the academic center, but not for the clinical site. So she would bounce around, but I guess she would be considered like a contract site organizer and
It might
everything.
a big academic center probably has
Yeah, she wasn't
coordinators part of
employed
their research. They
by
would come
them.
across, yeah, they'd come far, from across the street or in another building. Sometimes big hospital systems will actually outsource their whole research program
Okay.
for trials, and they might have been an SMO um, that consented you. That's fair game. They can do that. They just have to sign the 1572, all the obligations of the doctor. They have to have the oversight. All this is to FDA. It's allowed, but it's just a lot of layers.
Yeah. Oh,
The
that makes
true
sense.
CRO, the true contract research organization, their job is to kind of be an independent third party to verify that it's being done correctly. And their big job is they look at the EDC, they look at the electronic data capture. What did what site type in that the blood pressure was, that the side effects were this, that the patient was dosed on this day. And then they look at the other computer system and then they type in that system with the patient's records, right? They say, well, they don't type in.
Translating like as
They
a
look to make
project
sure
manager, I guess.
that source data, like, oh, the doctor did in fact dose the patient on that day. They did consent on that day. They did have a side effect on this day. So they make it so that the patient's electronic medical records are true as the source record, as to what goes in the electronic data capture, that goes to the FDA for the submission of the trial data. So those two systems are still very disparate systems. I say that's the vein of a lot of our investigators' existence.
Yeah, I mean, it's a lot of time to put in to do that. I mean, back in the day, you wouldn't have an electronic configuration. You'd be handwriting that out.
Back in the case report forms were paper-based. Now everything seems more modern because the technology is more modern in the sense that the EMRs are more modern, the
But you still gotta
EDC.
type it out.
So still today, I have 10 coordinators and they type in EDC every day. All the busy trials, everybody's typing in the data that they're seeing from our EMR, which is what our nurses and doctors put in the EMR records. That's the official medical legal record. But we can't put that in the EDC because they can only be the exact data that they only need for the trial outcome that goes to the FDA.
Can I ask this? Because I noticed this today because I was looking through some of my medical history, and it'll state on a lot of my records that it was voice recorded. So it was still essentially I in lieu of actually typing it out, it was voice recorded, but then it leaves a little disclaimer that says that it might not be 100% correct because of voice recording.
It gets worse. So, if when you have a voice recorder, that's the easy button for doctors. So sometimes they'll just I say blah
Because
blah
they're
blah.
busy.
They're busy. It's easier for them to have unstructured data, talk about whatever they think of that the patient just told them that they think matters, but they're probably missing some elements that the EDC might need for the trial. So then the coordinator will go back a week or two later. Doctor, you forgot to mention the performance status of the patient. Doctor, you forgot to
That
ask
explains
the
why
patient.
there's some addendums to some of
There's
those
always
records I was
trust me,
looking at.
the average number, like part of the CRO job, like the monitor visit, the CRA, when they come with the dolly and they stay for a day or two and they cross check,
Yeah.
they then find all the errors because they're paid
They're
for that.
like, "Oh my
We
god."
joke. They find the errors and they tell us if there's a deviation or we dosed a patient too late or we missed a urine sample. It's like the patient didn't have to pee. We still get dinged. It's okay. These things happen. A lot of it is things like something, a data point didn't quite exactly make sense. Like cancer patients, their heart rates are a little high sometimes. Okay. And there'll be a query, Dr. Powderly, why did your patient have a heart rate of 110? That's abnormal. Okay, yeah, we know that all my patients have a heart rate. Everybody's in pain. Everybody has a hard time. They're all anemic. They all have a heart rate. So that happens, but that leads to a query. So a query, it's a database term. It means you're getting a question, a query about why is the heart rate 110? That's above normal. So then there's a layer, I jokingly call them the database police.
What?
Sometimes I don't even know if it's a software creating a query or if it's an actual human that is a database police that's saying, I'm going to ask 100 times every patient that had a heart rate of 110, I'm going to ask, why is it 110? Or why is the heart rate elevated? We would do
I just felt like such a detective today as I was looking through everything because I was trying to line everything up and it was like, okay, I could tell that the information that was inputted was the time that they charted it, not at the actual time that it happened because it didn't coincide. And I was like, I'm trying to create like a correct timeline of my experience and all that and everything that went down. And it just didn't add up.
So you'll see the larger the hospital system, now you're going to get some of my Irish comments here.
It's
The
okay.
larger the hospital system, the busier the docs are in general. And then usually, and all doctors will agree with this, they get a little bit behind on their documentation.
They're always in catch up, I would
They're always in
imagine.
catch up. And so they get an administrative day, 1 day a week or a half day. And you'll see that they have all this administrative catch up on that day. And that's when the dictations go in.
Okay, so it's not
But
that
even that
explains why it's
4-day
not like
lag, it's easy to forget things, and so but what happens is sometimes they don't remember everything, and then they miss what might have been an important data point. Again, what's a pain scale? Come on, I forgot to ask the patient what number their pain is. Okay.
it's
Uh,
almost like the police, like, they almost need to wear, like, one of those cameras on their chest so they can just time lapse it and
there's
figure.
technology coming for that, and some of it's in use. There's all these things happening. Or the other thing is just ask the patient to enter their own data. Like that's another patient-reported outcomes or patient-reported outcome metrics. So these things are starting to have happen to make it a little bit easier, but the amount of queries are still a lot of queries. It's a lot of even though our clinic we are required. The reason we get to do such a good job and get good business is we enter all of our data within 24 to 48 hours. That means we as the docs,
That's
for
huge.
the coordinators to enter the data in the EDC, we as the docs have to enter our data on the day of or the morning the
Yeah,
next
you
morning.
can't wait for
You
like
can't,
your
because then
next
they have to enter
available
it.
moment or
And here's the other thing,
you
for us to have accurate data, I have found that it's a little bit of like teaching the new docs, and for us to have the most accurate data, we don't dictate. We don't dictate. We have templates. We will type very short, concise. The less you type, the more concise you are, and therefore, and then you hit what you
to
know
the
you need
point.
to mention. It makes it easier and more concise, and it's more structured. So even though we have an unstructured EMR, we're making it structured by the way we build our templates into it. So it'll at least be semi-structured. So when it gets ingested later into a more structured open EHR, it'll have the right fields.
So less detail at the time, but at least it's at the time. You can
Mhm.
add the detail to it later as long as you put the main bullet of
Ideally
what
in real
happened
time.
at that time.
The older you get, the more you forget. Oh, what did that patient just tell me? So
Yeah,
I have to have my laptop in front of me. I even tell them, dear patient, everything you tell me, don't talk fast. I'm typing everything you say so I can be accurate. Then you got to focus them in and say, okay, let's go back to what we started. Let's go back to this. I have a system. I have a system flight check.
makes sense.
I want to hear everything you got to say, but I got to go through these boring questions first.
I didn't mean to get off track, but I just had to ask that because I was
I
just
have
like,
my,
oh man, like it just rang the bell. I'm saying.
yeah, we have these style things in how we collect data so we don't miss things, which is another reason why we get a lot of good trials, is because we do have to ask a lot of boring questions to make sure we cover everything to get the full picture. So the next layer is the actual sponsor, right? The biopharma industry. You know, their business model is discover drugs, or now it's really engineer drugs. I joke with patients, we don't discover things anymore. We engineer things. And then it has to go in vitro, right, in a petri dish, and then into a mouse. And sometimes it might go into a rat or a rabbit or a beagle. And then usually into a monkey for final safety. And then it'll go to a drug manufacturer, but really drug companies don't manufacture their own drugs anymore. There's a whole other layer in there. I didn't even
That's where
put
the
the other.
CDMOs come
It's
in,
the
right?
CDMO layer, which is
Okay.
not even in here. They'll manufacture it for the drug company. And then after the trial is done, in that process, you know, that takes like 5 to 7 years. After the trial is done, it gets submitted to the FDA, and they look at survival curves and medians and statistics. And if the statistics and the curves look good according to the FDA's expectations, then a drug gets approved, right? So then you go from an IND, investigation new drug, to now an NDA, or a new drug application, with your statistics and your package, your 500-page package, that it can be approved for commercial use by the FDA based on that survival curve. So the deliverable there, the real deliverable of that whole phase 1, 2, 3 trial, at the end of the day, those top layers there are to make a survival curve, right? Right. So that you can show a median difference in the trend in the survival. And then the next layer is so that the drug company can hopefully have a blockbuster drug that works, and they can make enough return on their investment to pay for their $15 billion drug, because the top 3 layers are so horizontally disintegrated and based on the cost. So the bottom layer here is like the academics. Of course, academics are wonderfully important. We host students from all these academic centers, but their value proposition, at least back in 2025, is publish or perish. For publish or perish, you must, to get faculty tenure, you must publish, or you perish, meaning you might not make tenure. You might have to go get a job elsewhere or whatever. So they do a lot of research. They look for discovery targets. They even got early involved in cell and gene therapy. I like to say they have cured cancer in mice at least more than 200 ways. By now, we've probably cured cancer in mice thousands more ways. A lot easier to cure in a mouse. Mapping the genome, and it gets published in journals. I really should have put their deliverable, really their output besides text for publish or perish, is a patent. So that is the currency of an academic center. They don't, they can't have secret sauce like we might have. They can't really have business methodologies or efficiencies like we have, because there's so much bloat and oftentimes inefficiencies, or nimble or network model type things. So they do a lot of things good, but their currency at the end of the day is text, patents, discovery, teach, educate, do research trials at the academic centers. So those are inherently good things, but sometimes the outputs of all these don't really align oftentimes. And then the motive profit drivers also don't really align. And the reason I made this slide in particular, the way it's listed back in 2025, because I wanted to point out that the outcome in a in a simple way, the outcome of all these efforts at the end of the day, text, more text in an EDC, although it's a little more structured, a survival curve, right? Hopefully, a drug works.
Right.
Blockbuster drug, hopefully, it got you a blockbuster drug. And more text in the publications for academics. Great, that's good.
Can I ask real quick, is there
Yeah,
a
yeah.
specific criteria for those publications that academics have to publish to?
That's another discussion. I mean, there's basically back then and still today, if you are a good grant funded research professor and have a lab, you're doing good enough to survive, but you're not a millionaire. And you're up till 11 p.m. writing grants. This is well known.
Yeah.
They work hard. It is a tough but important job. And there's now more and more journals are coming out. There's a journal for everything because the knowledge base is getting so big.
I was gonna ask.
China now has more journals and more publications than we do, like in our heartbeat. And the grant funding is getting worse and worse. I can't make this up, but our chief scientist just told me an hour ago that on the news, the entire board of the National Science Foundation was let go. I haven't verified that, but he saw that. I'm like, oh
I'll have
my
to
God,
check the evening news later
that's
on,
unbelievable because they
but
already cut a
that
lot of
that
the
blows my mind.
NIH. They now that's like the entire National Science Foundation, which is some of our best spent money. So the academics are dependent on state and federal government
Uh,
for large part. So, and that's where the early discovery, some of the best technology is born out of. So that's hard. So that layer is
well,
under
that hurts,
stress.
man. That hurts. I'm a steering committee member that
So
is a part of that. So
you
oh,
need to hear
dude,
about, yeah. So that's what, according to him, not to vet that, but
that's
that's
okay. That's okay.
so. All right.
That's
So back in 2005, this was the slide, but why we wanted to build the model to be more granular, elemental, or relational database that could compute instead of just the text does not compute text. So we could build this model. So, and I started on the left. First, you got to have a tumor bank, right? And not just a paraffin wax tumor bank, but
yeah,
actually
not
like
just
a living
seven
tumor.
plus year old slides sitting in
Bingo.
a
So
drawer
we back in 2000, we started a living tumor bank.
in a freezer, right?
Well, liquid nitrogen and -80.
Yeah.
But if somebody had a biopsy, we would take a sliver with the good graces of the hospital system in the IRB approval, and it's a consent study. We would call it our laboratory trial, right? Patients would come here for a phase 1 study. Great, but dear patient, we're doing our own research down the hallway. We have a laboratory study. We want your blood. We're vampires.
Yeah.
Want extra tubes of your blood, and we want a piece of your tumor. You're going to get a biopsy anyways. You're
I've
due
done
for
that
see,
and I've
we'll take a
contributed
little
to that.
sliver as that we
Absolutely.
joke with the patients. Dear patient, everybody wants a little piece of you. The hospital needs a paraffin wax. The drug sponsor needs their baseline biopsy for
Yeah.
their
If you
arm.
got enough to spare,
There's
I
an
mean,
extra
everybody can get
sliver,
a
we'll
chunk.
grow the tumor. So that was the model, and then we would look at genes or this is kind of going in the order of complexity across the top. Genes, proteins, metabolomics, like how do proteins interact, signals and metabolism. Cellomics, how do cells interact, but cells are doing the work, and then they're taking cells together and battlefields, immunomics. So does your immune cell kill or not, or what type of immune cell kill? It's getting into the fractions we're working on, and then the clinical data. So that's why from day one, we never dictated into a dictaphone. We just always would put our data as very much like an EDC, but put it in an EMR as the most granular data elements that we could. And the reason there is as we would grow over 20 years and we have all this data, we would then do what we would, back then, these were the buzzwords, in silico medicine, looking at your relational database for down on the left there, hierarchical clustering, seeing things that relate through clustering analysis. Running the computers would run all night and look for what things relate at artificial neural networks. These are other software programs that you could run, self-organizing maps, circuit networks, and to basically look at that system. Now we know that as AI, modern AI, this led to AI, and now we're kind of hearing like if we trust AI in a regulated environment, we're kind of good machine learning practices or GMLP, like you've heard GCP, GMP, good clinical practices. So
Okay.
for machine learning, if it's very regulated and if the FDA is willing to hopefully trust it, if it's got the barrier, the guardrails on it, AI is being used for some diagnostics, radiology, mammograms, even some pathology stuff now, clinical decision support for doctors to kind of get hinted at, oh, think of this and that. So that's good machine learning practices, which means the accurate data in should be structured. It's got all the security around it. Doctors are supervising it. So that was the model of the why back in 2005. So I would show this slide to the docs at this conference, say, hey, this is what we're building. This is why it's important. If you guys do trials, you could do something similar. We're happy to work with you, try to set up a network. So then the next slide, and then we'll be another 4 or 5 minutes and we'll break. So then this is the why, again, is what we all learned about the biopharma industry. Great, it works. We need blockbuster drugs. Yeah. And the FDA looks at it, their deliverable, the FDA expectations is it's a process you have to define every step, validate every step to make a product. And so they say that the process is the product, right? And there's critical quality attributes that you have to test and verify, highly regulated, really great ways to think about a process makes a product for a blockbuster drug, one size fits all. So we're saying, okay, we understand that paradigm as the drug paradigm, blockbuster drug. Great. 1990s, year 2000. Here's, Dr. Powderly. What if we want to create a cell just for one patient, like Dr. Rosenberg? What if my mentor, he taught me how to do that 35 years ago? What if we want to do that? How can we think of a business model that still satisfies the FDA, that is a process to make a product, right? But not in as a vial of one size fits all, but really the product is a cellular product down the hallway for that one patient, a single batch. And we want to give it to them at the same place that they came in. It has the clinical door, and it's also the manufacturing all in one place. So it's a huge paradigm shift. So yes, that was the model 21 years ago. So in that we would be delivering the process as a product, but we're delivering it as a professional service, right?
Exactly. And we've talked about this
Yeah, so
multiple
that's sort of
times.
the whole model. So it's really cool. So this was like a lot of the different devices, tabletop devices. That was the reason why, and it's fun to tell the docs about this. So next is why to build it, and maybe we'll pause here. So then when we start the next talk, we can kind of start off with the why the doctors. Why would you want to do this?
Most stage 4 metastatic solid tumor patients are still incurable.
That's a why.
Oh,
That's a
dude.
yeah. This is
Yeah,
gonna be
this
good.
slide just hits home. I
Because
mean,
then we're going to start getting into from the doc's perspective. All right, would you do this? If the patients hear about it, they would do it. But dear doc, now that you're hearing about it, would you do this?
uh,
What's your bandwidth? What's your interest? So
exactly.
we'll stop here and then we'll start up again in another couple weeks. And
Absolutely. Wow. Again, Dr. P, what a great session today. What a great slide to end on. I mean, it's like a cliffhanger. I
Chris,
really like it's really getting super exciting.
we haven't even got into all the insurance. Like there's data on insurance denials for trials. There's data on how many doctors sign up to be an investigator, one and done. They're like, I'm never doing this again. They don't
Yeah,
want the regulatory burden. I mean, there's a lot of data that I'm pulling out from different resources that paint a picture of how tough the research system is in America.
it's hard. And the conversation about scalability never ends. I hear it everywhere.
Yep.
So
Yeah.
this is be a great one to pick up on for our next one. To everybody out there, thank you for joining in and sending love. Peace.
All right, that concludes today's episode of TIL Talks with Chris White as we talk TILs, tumor-infiltrating lymphocytes, and all advanced cell and gene therapies. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. I'd like to thank my guest today for joining me on TIL Talks. To our listeners who have not yet done so already, please follow, comment, subscribe, share, and smash that like button. For more information about TIL Talks and related resources, please visit www.tiltalks.com. I'm Chris White. As always, sending love to everybody. Peace.